So yes, we're going to go into the same level with nerves to indicate how nerves work and then how they drive muscle, and then we're going to put them all together with the nerve functioning, with the muscle functioning, and show from start to finish, from signaling from the brain basically to contraction of the muscle. Work all the way through. So we've seen nervous tissue before, now we're going to go into more depth. So here's another example of a nerve. There's a new term here, perikaryon. Don't worry about it. It simply means the cell body, not including the nucleus or the axons or the dendrites. It's just a very specific term for what's really contained in the cytosol. And that's it. So I won't be using it, but it just happened to be on the image, so I didn't want you to be confused by it. However, you should be familiar with already the dendrites, which are the processes that bring information into the cell. The axon, which is the process that takes the information out of the nerve cell. And something new that we're going to focus a bit on here is the axon hillock, where the perikaryon meets the axon. So where the rest of the cell body, it's a transition zone between the rest of the cell body and a very highly specialized axon. And this is just an arc representation of the same thing. So once again, you get a single cell, doing what all single cells have to do, what every single cell has to do, maintaining homeostasis. It's got help, it's got those astrocytes helping it, a bunch of other cells in that tissue type. You have the cell body, you have the axon, and the axon hillock. But I really want you to take notice of now, this is now going to become quite important, where I just kind of glanced over them when we talked about nerves at the tissue level. It's these myelinated Schwann cells that wrap around the axon and the spaces in between. So how a nerve works and how it transmits an action potential, how it transmits signal, is very much dependent on this system. So we're going to look at that also in a lot more depth. And then of course, we'll end with what happens here. Axon terminus, which is how it transmits that signal to either another nerve cell, or a muscle cell, or a gland, or whatever. We're going to be talking quite a bit about the transmitting of charge. So that's an electrical charge, positive and negative, current, mostly negative. But it's really important that you get your head around the notion of what a charge is, and we'll be talking about that in a minute, but just to set this up. When electrons are added or removed from an atom, it changes the charge, but it's not intuitive. When you add electrons, you would think it'd be more positive, because adding is a positive thing, but that's not the case. If you add electrons to an atom or molecule, it becomes negative. If you remove electrons, it becomes positive. So it's not quite intuitive, but remember that. So once again, we're going to start with some terms, of course, and then from those terms, hopefully walk through the whole system. So we need to know what ions are. I've mentioned ions before. I've talked about ion concentration, ion balance within a cell. Haven't really defined what an ion is, so now we're going to. It's an atom or a molecule that has a charge, so it's either negative or positive. It is not neutral. There's lots of neutral particles, but ionic simply means it has a charge, so it's either lost electrons or it's gained. Is everyone familiar with an atom and the atom structure? So I can draw that out if you wish, but anybody want that up on the board? No. But atoms are just made of protons, neutrons, and electrons. As long as your electrons and protons are in balance, there's no charge. It doesn't cost a penny. If you remove electrons and there's not the same number of electrons and protons, either negative or positive charge, depending on if there's less electrons or less protons. So it comes from the gain or loss of electrons to be ionic, and so the electron number does not equal the proton number. We won't have to talk too much about electrons, but the concept of an ion is very important. cation is positively charged, so it's lost electrons. So yesterday when I was talking about calcium, you notice that I had a C representing the letter, the atomic letter N++, meaning it's lost 2 electrons. So it has a double positive charge, or 2 more electrons, sorry, 2 less electrons than it has protons. So a cation is one that's positively charged. An anion is negatively charged. So I will be using these kind of interchangeably. Sometimes I'll just refer to a positively charged ion by its name, but if I don't, I'm saying, well, the cation crosses here, balancing the anions, then I want you to have that straight in your brain. So this is positively charged, negatively charged. And this is a really important concept to understand the action potential and how these things work. This whole notion of concentration gradient. Nature abhors imbalance, so it does whatever it can to put things equal. And body systems use that to passively move things. So whenever there's more of a substance on one side of the barrier than there is on the other, you have a concentration gradient. So let's take cells as an example. So we have, there's a cell wall, let's say. There's a cell wall, we got a plasma membrane, and a bunch of cells here. And up here is the ECM, which is your extracellular matrix or your extracellular fluid. So all I'm saying here, in terms of ions, let's say we have a bunch of cations. And there's a, in terms of a ratio, there's some here, but there's lots in here. Let's say there's just well, there's a concentration gradient here, and the tendency would be moved this way. If those cations could move through that plasma membrane, they would go to the cell into the extracellular fluid until there was a balance. Okay? So there'd always be movement, but there'd be no net movement. I mean, overall, you'd have the same on either side. So cells purposely set up these gradients so that they can function. And we're going to be looking at 2 kinds of concentration gradients, and some of the times at the same time. That can get a little confusing. We're going to be looking at chemical. So in that case, I would say, maybe we've got some sodium ions here. Sodium, maybe it's a balanced situation. So that's going to be chemical concentration and electrical, which I've already shown you. And of course, then you can see them at the same time. So there can be an imbalance across the cell membrane, both electrically and chemically, and sometimes in opposite directions, which creates some interesting transport. So keep that straight. Concentration gradients, more or less of something on either side of the barrier, and they can be either chemical or electrical. An electrochemical gradient, as I just mentioned, is a concentration that is an imbalance of both the chemical, or the molecule, and the charge. Motor neuron, that's what we're going to be dealing with mostly. It's a neuron that carries information to an effector cell. An effector cell is just the cell that does something. So in this case, the effector cell is the muscle cell that contracts. It could also be a gland that when motor neuron stimulated, it stimulates the gland to release a hormone or what have you, or sweat gland. But there are 2 other kinds, two other kinds of neurons. Excuse me. One is for sensory input, so that's the information going from your eyes or your skin or your taste buds to send information into the central processing area of your brain to have that sensory information to process and send back and out. So those are sensory. And also for interneuron communication, where one neuron is sending information to another. Those are your 3 basic types. Interneuron is nerve to nerve. Sensory is special neurons to central processing in the brain, so we can interpret what we're seeing. And motor neurons do something. They affect change somewhere else. But that's what we're going to be concentrating on. Effectors, which I've just mentioned, is the tissue or the cell or the organ that it is impacted or responds to the signal from the motor neuron. In this case, it's going to be skeletal muscle. We looked at. And that nerve impulse can either be electrical or chemical. So what a nerve impulse is, in a nutshell, is a change in the electron gradient across the membrane that travels down an axon. I realize it. I'll explain that later. But that traveling down an axon is how the signal is actually sent. And that change in the electrochemical gradient is the potential moving all the way down. So all nerve information is either electrical or chemical, depending on the nerve and depending on the effector. Intranerve stuff is all electrical. Heart muscle is electrical. But as we're going to see, when motor neurons to stimulate skeletal muscle, it's actually chemical. It starts out electrical in the neuron, but the transfer of information between the neuron and the muscle cell is chemical. A couple other terms before we can get into the explanation proper. Resting potential. This is when a nerve is not sending a signal. This is resting. So there's nothing going on along the axon. All animal cells have a net negative charge along their plasma membrane at resting potential. So right at the plasma membrane, it is slightly negative compared to the extracellular matrix around it. That's normal. Not so sure about plants, but all animal cells, especially all animal nerve cells. And this is due to the presence of sodium. Because the animal cell is constantly removing sodium. Sodium is necessary for certain functions. However, high concentration in the cell, highly toxic. So your cells have specialized pumps that remove the sodium at a rate higher than it's coming in. And there's also some chlorine ions that are already inherently negative. So this creates a slightly negative environment in the cytoplasm of the cell and along the plasma membrane compared to the extracellular fluid. And everything is comparatory here. That's really important. Like, if you say something's negative, it has to be negative compared to something. Or if it's positive, it has to be positive compared to something. So some terms we like to use. We have hypertonic. Hypertonic is where one region, whatever it is that you're talking about, has more of something than another. So I have all of these pluses here. Let's add some sodium as well. So this cell is hypertonic compared to this environment. It's got more sodium than this one does. Whenever you use a term like these, they're always relational. You can't say the cell's hypertonic. It has to be hypertonic compared to something else. So this cell is hypertonic compared to the outside environment. The outside environment is hypotonic compared to the cell, meaning there's less. Hypertonic, hyper, think more. Hypo, think under, so less. So the extracellular matrix in this example is hypotonic compared to the cell. Then there's isotonic, that means spelling right, which is equal, but there's no net movement. There's the same amount of sodium on both sides of the cell. So in that case, this cell, let's say, is isotonic compared to the environment it's in. It's got the same amount of sodium as the outside does. Now that doesn't mean there's no net movement across the membrane. There still is, but there isn't a gradient. So over the long term, it stays balanced. And the fourth one, the most important, gin and tonic. Absolutely, my absolute favorite of all the tonics. That's a joke. You don't have to know gin and tonic. Though in the summer on a hot day, very nice. So the resting potential is nothing's going on in the cell. Perfectly fine. And there is a net negative charge in the cell compared to the outside environment. Action potential is the signaling, is the nerve cell firing. That's what action potential means. It's one of those words that, once again, is kind of misleading because it's not a potential, it's an actuality. So when you say action potential, you're like, something's about to happen. Like, no, it's happening. I don't know, why do they do that? It should be just called action. Stop. Because it's the cell firing. It's that nerve actually transmitting information. And it's a brief change in charge, because there's that nerve cell receiving a change in charge from the one before it, and it's going to pass that on. But the whole nerve doesn't change at once. Otherwise, it would be firing constantly. It changes in increments all the way down the axon, ever so slowly, in a cascade event we will look at. So the cytoplasm in the axon changes. It's from the slightly negative to a slightly positive. And that causes things to happen, all kinds of interesting things to happen, which pass that action potential down the axon to the next neuron or to the effector cell. So the extracellular environment around the cell changes from a net positive to a net negative, but only for a short period of time. So this moving of the electrical current along the axon is the action potential. That may not be perfectly clear yet, but hopefully it will be by the time we're done. But for now, just know that the action potential is the actual signal, the movement of electrical current along an axon. Channels. Channels are the gates that we talked about, I've seen now forever ago, I'm sure, where we looked at the plasma membrane, city walls of our cell, and we have ways through them, openings that are specific to particular molecules. They're also called channels. And the protein molecules that embed in the plasma membrane that go all the way through. So part of that protein molecule is in the cell itself, and part of it is outside the cell, in the intracellular matrix. There are loads of these along the axon at very specific places. They're in the nodes of Ranvier, because the nodes of Ranvier is the only place the axon is exposed to the extracellular environment. The rest of it is wrapped in a myelinated sheath produced by Schwann cells. So the only place that you can have an exchange of either electrical current or chemicals is in those nodes. So a channel is just protein specific to a particular molecule allowing things in or allowing them out, or both. A gated channel is one that is normally closed, and something specific has to happen to make it open. Some are activated by ATP, which we now know all about, but some aren't. Some are activated by chemical messengers, neurotransmitters, which we'll talk about later. And some of them are activated by voltage. So that change in electrical current changes the shape of the protein, and then it opens up and allows something else to happen. But the point of the term is that they're normally closed unless a specific event takes place. Either ATP shows up to open it, it's that kind of gate. There's an electrical current change, if it's that kind of gate, or a particular chemical shows up, if it's that kind of gate. And I will let you know which ones they are as we go through. But very important to know the term voltage gates, because we're going to look at a lot of those. They drive the whole action potential. A particular one is this, and this is very important. All cells have it, especially nerve cells, muscle cells. It's the sodium-potassium pump. This is a particular gate that's constantly functioning and does two things at the same time. It removes sodium from the cell at a rate of 3 sodium molecules and brings potassium into the cell at a rate of 2. So for every 3 sodium that leaves, it brings in 2 potassium. They're both positively charged. But it creates a net negative charge because you're losing more positive charge than you're bringing in. Right? So it's a net -1 for every event, if you will. So you're losing 3 sodium, which is 3 positives, but only getting 2 back. So what this does is it slowly, over time, creates a net negative charge within the cell. That's why animal cells have a slight net negative charge. K stands for potassium, by the way, in case you weren't sure of that. I'm not sure why they picked K, but they did. Because P was already taken for phosphorus, actually, so they had to come up with something else. But both are positively charged, however, there's this net negative because you're bringing in less than you're sending out. So those are always functioning. Well, mostly always. Almost done with the terms here. I've mentioned this now a couple of times. Now we focus on the axon hillock inside of a nerve cell. We'll take a look at it. It is, I say, it's a transition area in the cytoplasm of the cell, transitioning from the rest of the body of the cell to that very specific axon. And what it does is it has a bunch of sodium and potassium gates associated with it, and it receives the charge from the dendrites, so an electrical current coming in. But not just any electrical current is going to activate it. There's a certain threshold level. You have to have so much before those proteins change shape, start opening things up. But once a certain threshold level has been reached at the axon hillock, it starts the whole process that sends the potential down the axon. So basically, think of it as a light switch. It's off until some threshold of electrical current has been reached, and flicks on. As soon as that happens, a whole bunch of things cascade after that. So this collection, if you will, of positive charge collects in the axon until there's enough of a positive charge to open up the voltage, the sodium voltage gates, starting the cascade of an action potential. But don't worry, I won't explain all this again. I just want you to know the terms first, and then we'll take a look at it. We'll walk through the whole process. So looking back, here we have a nerve cell again. We have the dendrites. Attaching to these dendrites are the terminus of other nerve cells. So this is the information coming into the cell. You have that information gathering and collecting. So basically, this is a net negative cell. All of that information is making it slightly more positive. Every time there's a signal, slightly more positive. And that positive electrochemical gradient builds up here until it reaches a point that is positive enough. It opens up the gates there, and the signal comes up down the axon. Now this happens much faster than I'm explaining it. Of course, it's milliseconds. And finally, the refractory period, which is a period where the cell cannot fire after it's gone through this entire process of charge, depolarization, where it goes from slight negative to slight positive, and then back, repolarization, and then it has to rest. It cannot fire again, no matter how much information is coming to it from other dendrites, from other neurons, axons, it won't fire. And that's not a very long period. It could be, once again, milliseconds. But this prevents the nerve from being constantly on. So it's a safeguard, so it doesn't burn out. So, now that we have the terms down, let's walk through slowly how this whole thing works. So, draw this out. Once again, you'll be stuck with my great artistry. We'll also watch a flash animation. Right? Actually, I have a graphic one. It's quite nice. So here we have our nerve cell. I'm going to draw a couple of dendrites here. Sometimes there's a lot more than this. And our one very long axon. Nucleus, got to have a nucleus. Organelles are in here, but I want to focus on is this bit right here. This is our axon hillock. Okay. So, this is a slight negative environment compared to a slight positive environment around it. Right? Information's coming in. Electrical current from another nerve cell, which is slightly positive. So that's going to make this light negative, less negative, if you will, to slightly positive. Once it's positive enough at the axon hillock, that threshold has been reached on the switch. This is called depolarization. And what you have here, so right around here, you've got, there's more than two, trust me, you've got a whack of sodium pumps that have up to this point have been closed. Right? They're a voltage gate, sodium pumps. Now remember, the whole time, the sodium-potassium pumps have been working to remove sodium. So there is a net gradient of more sodium on the outside and less sodium on the inside of the cell. This is before it fires. This is resting potential, right? That's important to know too. So we have both a chemical, there's more sodium on the outside than on the inside, and electrical, positive on the outside, negative on the inside gradient. So the natural tendency, if there was nothing stopping it, is to have sodium and positive charge ions rushing to that cell because there's less there. And that's exactly what happens when the axon hillock is activated. These sodium pumps open up. So this positive charge changes the shape of those proteins enough that they open up and sodium just rushes in. Which causes something to happen. So now the area around here is much more positive, which makes the area right next to it more positive compared to the outside. And exactly the same thing happens. All the sodium pumps here, they open up and sodium rushes in. The key to remember here is it isn't happening all at once along the axon. It happens in stages. So it happens here, because remember this area is still negative compared to the external environment. It happens here, which causes this area to become positive compared to the environment. And its sodium pumps open up and sodium rushes in. So you have a slow, well, not slow, it's very fast, but you have a gradual movement along the entire axon of going from a net negative charge to a net positive charge because the area next to it did. So as the area next to it becomes positive, that positive charge opens up the sodium voltage gates right next to it, allows sodium in, which makes that area positive, which opens up the sodium voltage gates next to it, and so on and so forth, all the way down the axon. That way that current gets moved down the axon. Does that make sense? And that's really important to understand that. Any questions about that? Let's talk right here for a minute. No? Okay. And in terms of the actual difference, most animal cells start at about minus 70 millivolts. That's just, if you measure that negative charge, that's what it measures as. When it depolarizes, it goes to minus 50. So it's still negative, it's just less negative and positive in relation to the outside environment. So what this is, this isn't a cell. I say that because I had a student enter this on an exam and they thought it was the cells and started labeling this. They didn't do very well, I can show you. This is a graph, not a cell. It's a graph of what's happening in terms of the action potential. So this is measuring millivolts. So what are we going to do here so far? So we start here at resting potential before anything has happened. signal comes in, and we're at, this says minus 90, but I guess it varies. Anywhere between minus 70 and minus 90, but don't worry about it. So this particular nerve cell, its resting potential is minus 90 millivolts. You start getting positive charge coming in from the dendrites. And that's what this is. It's slowly accumulating in the axon hillock. But nothing is happening yet until enough of it's there to trigger the axon hillock to open up its sodium channels. And that's what happens right here. That's your threshold level. So all of a sudden, you get a slow build up in the axon hillock. Threshold levels reach, sodium gates open. And then the next sodium gates open, the next sodium gates open, and then you have all the sodium rushing in. So what this is saying, it is taking about 2 milliseconds for that action potential to travel all the way down the axon. So right now, right here, it's at the axon hillock. It hasn't gone any further. Right here, it's done. So measure that, not even 2. It's 1 millisecond to do that. But the point is, it takes time, and it happens in a stepwise fashion. Not all the gates along the action potential open at once. So only once the positive charge reaches a gate, it opens, makes that area less negative. The next gate's open, makes that area less negative. The next gate's open, all the way down. So we're going to stop there. I haven't told you what happens next. So this is the action potential shooting down the axon terminus. That's just showing you how fast it happens and the change in voltage. So we've gone from minus 90 at around minus 65 is the threshold for this particular cell. And then within a millisecond, it goes from minus 65 millivolts to plus 35 millivolts. That's how positive the environment now is inside that axon. You don't need to know these numbers because they vary. As I said, most animal cells they start at minus 70. Just know the relation. You go from negative to slightly less negative at a threshold to actually positive. And that is depolarization. That's it's going from a net negative charge inside the cytoplasm along the membrane to a net positive charge. This is how this becomes depolarization, and the movement of that depolarization along the axon is the action potential. Any questions on just that so far? I know that can be really. And the reason that it works is because the change in charge, negative to positive, opens the voltage gates, meaning it changes the protein shape so that it allows the sodium to come in. So it further changes the protein shape. So now we have a situation where there's a ton of sodium in the cell. Because with the sodium comes the positive ions. Should say come with positive ion. The sodium is the positive ion. And as I said, this is going to work its way all the way down the axon. But now we're left in a situation we have a positively charged cytoplasm, which is great for if you only want to fire once, but not so great if you ever want to fire again. And so what happens is 1 millisecond after those gates open, and allow all that sodium in one region, gates next to it that pump potassium out open. And the delay of 1 millisecond is very important, because otherwise, if they all opened at once, there would be no net change in charge, because you'd have positive going in, positive going out, so there'd be no action potential. So here we have, here, potassium gates now open. They also open because of the change in charge, but instead of pumping potassium in, there's already more potassium inside the cell, because it produces a lot. More potassium inside the cell than there is outside the cell. So now, because remember the Sodium potassium pump has been pumping out sodium all the time and bringing in potassium, and that's an ATP-driven pump. So it can bring potassium in against the concentration gradient. So there's less potassium outside the cell than inside, but still the cell can bring it in, and it's applying energy. So potassium rushes out. Potassium goes out of these potassium gates and open 1 millisecond after the sodium gates open. So now you have an exit, this massive exit of sodium, of positive ion. So this is going to start that process of bringing it from a positive charge back to a negative charge. And within that millisecond, you'll have up to 7,000 ions crossing. So when the sodium gates open, in that millisecond, you'll have up to 7,000 sodium ions crossing at a single gate. And when the potassium gates open, 7,000 of those moving out. This is called repolarization, because once again, it happens in a stepwise fashion. So the potassium coming out here changes this back to a net negative charge compared to the outside environment. What that does is it closes the sodium gates because they're no longer receiving the right electrical current. The gates change shape again to the original configuration and close. And the potassium gates open here, and the potassium gates open here, and you get that repolarization going down the axon in exactly the same stepwise fashion as the action potential did. And since there's no positive charge, all of those proteins and gates go back to their normal shape, and there's no more ions passing, except in the potassium sodium pump, which is constantly going, constantly making the internal cell a little negative. So going back to the exact same graphic, we'll continue where we left off. So we have an action potential at the axon hillock. Depolarization takes place, sodium gates are opening within a millisecond per gate, you get 7,000 sodium ions pouring in. We get to here, a millisecond after that happens, the potassium gates open and start pumping potassium out. So while at the axon hillock, while potassium is now moving out, we still have that action potential moving further on down that axon. So it's both happening at the same time. So now we have positive charge leaving, so it starts to get more negative and more negative and more negative and more negative until you get to the point that it actually goes below where it normally is. This is called hyperpolarization. And when hyperpolarization is reached, the potassium gates close. And then it's just a matter of the sodium and potassium pumps doing their thing. And that's the end of your action potential. That's one signal that's just been fired. So we've gone from resting potential to action potential, which is depolarization going down the axon, to repolarization. Does this make sense to you? This is kind of important for you to get this. How quickly it happens and the change in charge. So this is the action potential happening. I say, and all along, meanwhile, the sodium potassium pumps continue to move sodium out and potassium in. That's how you get that constant, that's how you reestablish the concentration gradient in the cell. Because remember, during repolarization, the cell is pumping out potassium. So it has to have a way to get all that potassium back. So when the next action potential happens, there's more potassium in the cell than there is outside the cell. And there's more sodium outside the cell than there is inside the cell. Because if there was an equal amount of sodium in the cell than outside, when they opened up, no sodium would rush in. There's already a balance. The reason it rushes in is because there's less sodium inside, and there's a negative charge inside. So that's maintained by these sodium-potassium pumps. And I'd say that cycle of depolarization and repolarization is the action potential. So the key thing to keep in mind is there's two things happening at once. There's a change in chemical gradient and a change in electrical at the same time. I mentioned before, once one action potential is complete, there's a refractory period where the sodium gates cannot open. And all of this takes place at those nodes of random neuron I mentioned yesterday, in between the myelinated cells. So it cannot happen everywhere along the axon, because most of the axon is actually covered up in these myelinated sheaths. But there are gaps in between them, we saw in the first image, I think. And those gaps are called the nodes of Ranvier, and that's where all of this takes place. Finally, we're going to talk about in a bit. First, I want to show you a flash of what we just talked about to make sure that's settled. But then we're going to talk about, but I haven't drawn it yet on purpose. We're going to talk about what happens here at the axon terminus. Because once the action potential reaches here, the very end of it, how it transfers to the next cell depends on what kind of cell it is and what kind of nerve this is. So we're going to assume this is a motor nerve, it's going to go to a muscle cell. So the transfer of that action potential goes from electrical to chemical. And that process is really neat. First, let me show this video. Hopefully it will cement what I just said and make more sense. At least more sense than my drawing. Hopefully I made sense.
This baseball player springs into action, his nervous system rapidly processing information in a network of nerve cells or neurons. Each neuron receives input from one or more cells. In response, the neuron may generate an electrical signal known as an action potential that travels down the length of the axon. Let's take a closer look at the plasma membrane of an axon. Even without an action potential, the axon is a busy place with many ions moving across its membrane. Much of this ion movement is driven by the sodium-potassium pump. Using energy from ATP, sodium-potassium pumps actively transport sodium ions out of the cell and potassium ions in, creating an uneven distribution of charge across the membrane. Some potassium channels are open all the time, allowing potassium ions to leave the cell. As a result of these ion movements, the inside of the cell is negative relative to the outside. This condition is called the resting potential. The membrane of an axon is also packed with gated ion channels that open and close during an action potential. At resting potential, the gated channels are closed. If a stimulus changes the distribution of charge across the membrane sufficiently, the gated sodium channels open. Movement of sodium ions across the membrane makes the inside of the cell more positive. This reversal of the charge distribution causes the gated sodium channels to close and the gated potassium channels to open. As potassium ions move out of the cell, the original charge difference is re-established across the membrane, closing the gated potassium channels. This sequence of events is called an action potential. The sodium-potassium pump restores the distribution of ions back to their previous levels at resting potential. Let's look at how the action potential is conducted along the axon. As the change in charge difference across the membrane spreads from open sodium channels, other sodium channels farther along the axon begin to open. The original sodium channels close, and adjacent potassium channels open. As potassium ions move out of the cell, the original charge difference across the membrane is restored, and then the potassium channels close. Meanwhile, new sodium channels open, followed by the opening of new potassium channels and the closing of sodium channels. In this manner, the action potential is propagated along the axon of the neuron, eventually reaching another cell. The information carried by this action potential will be processed with other information, permitting this baseball player to make a spectacular catch.
Any questions so far? Okay, so that's basically how nerves work. We've looked at how muscle works. Now we're going to put it together. How does a nerve drive a muscle? So we're only missing one piece of information. We know how muscles contract. We know how nerves send signals. But how does the signal get from the nerve to the muscle? How does the muscle interpret it? And this is different for different kinds of effector cells. So remember, the effector cell is the one that's responding to the signal from a nerve. Different kinds of cells respond, well, since they don't all respond uniquely, they either respond electrically or chemically. So, excitation, contraction, coupling. This is just a term for how a nerve drives a muscle cell. And this is kind of the take-home message. This is what I want you to be able to explain to me, start to finish this whole process. So it's the complete process from the nerve firing, from the axon hillock of one nerve to a contracting muscle cell, which includes the cross bridge at the sacromere that we talked about yesterday. So a couple new terms. Neuromuscular junction. This is where the axon of a motor neuron meets the sarcolemma, the plasma membrane of a muscle cell. So there's an interface there. That is called the neuromuscular junction, which makes sense. It's the neuron and the muscle. Specifically, and I will draw this out for you. The motor end plate is the area of the muscle cell at the neuromuscular junction. And it has its own name because the structure of it is slightly different than the rest of the sarcolemma. It's heavily folded, so we increase the surface area with lots of sodium pumps. And we know how important those are for changing the current. Because what's going to happen is you have an electrical current gets converted to a chemical current, gets reconverted back into an electrical current. So electrical in the nerve, chemical, the interface between the nerve and the muscle cell, and then becomes electrical again in the muscle cell. Couple more terms here. The synaptic cleft. This is the space between the neuron and the effector cell. They don't physically touch. There's actually a gap. It's really small. It's like 10 to 20 nanometers wide, tiny. Can't draw a car through it. The electrical synapse. This is action potential that's being directly transmitted electrically. This happens in cardiac muscle. That's why it can be uniformly. It's faster than the neurotransmitters. It's faster than chemical. It's not the one we want to be examining. It's one to be aware of. That's the other one. So this would be, instead of going from electrical to chemical back to electrical, it's just electrical to electrical. That's how neurons interact with each other. And obviously, some effectors. Chemical synapse, the one we're going to be examining, is where the action potential from the neuron opens up calcium. Once again, calcium cations. Once again, showing how important calcium is. So they open up, and I'll draw this out in a minute. They open up the action potential along the axon. When it reaches the end of the axon, at the end of the axon, you have calcium channels, all also voltage controlled, like the sodium channels were voltage controlled, the potassium channels are voltage controlled. These particular calcium channels are also voltage controlled. And there's a gradient, there's more calcium outside the cell than inside the cell. The action potential reaches the end of the axon. That change in current, slightly negative to slightly positive, opens the calcium channels and allows calcium into the axon terminus. In the axon terminus, in the all-important vesicles, that we have those vesicles, transport thing, is acetylcholine, which is a neurotransmitter. And it's not interacting with the rest of the, not interacting with the rest of the neuron because it's in vesicles. And in short, what happens is the calcium comes into the axon terminus, binds with those vesicles, guide it to the plasma membrane of the nerve cell, where the vesicle membrane binds with the nerve cell membrane, and that acetylcholine is released into the synaptic cleft, into the space outside the cell. And then something happens. It affects the muscle cell, but we'll walk through that better, that explanation. But for now, I just want you to be aware that there's an electrical synapse for translating electrical current to electrical current, and a chemical synapse which translates electrical to chemical, and ultimately back to electrical. So in this diagram, don't worry about nerve A and nerve B. All I want you to see here is this is the interface. So this is a big picture look at it. So we're looking at this as multiple cells. So each one of those is a single cell. So inside one of the cells, you see a bunch of the myofibrils. So we're looking at a tissue level organization. This is where individual nerves are now connecting to muscle cells. So where they connect right here, that's your neuromuscular junction. That's kind of a big scale picture. Well, big compared to the other scales I'm going to show you. So the action potential is generated in the neuron, right? Moves along the axon. So I'm going to draw this. So I'm going to have to walk back a bit. So here we have the axon terminus. It doesn't actually look like this, I'm just going to draw it like that. So remember, what I'm drawing here is the boundary of the cell. This is pretty much the plasma membrane I'm drawing. So we had all these sodium, potassium gates all along here. This we have calcium gates. Also voltage gates, voltage driven. And they're all over here. Also, you have within vesicles, we'll make pink for the fun of it. Inside of here, sort of sitting inertly, closed off, filled with acetylcholine, which would be yellow, which is a neurotransmitter. Without anything else happening, they will just continue to sit there. But something does happen. We get our electrical current coming down. So now we have a net positive charge here compared to a net negative charge there instead of the reverse. And this opens up the gates. So sodium rushes in. Are you with me so far? Those sodium ions come in and they attach, sorry, not sodium, calcium, I keep doing that. Calcium ions attach to these vesicles. I don't know how many ions have to attach to an individual vesicle. I suspect it's more than 1 to 1, but I don't know that. That's not the point. And then they move them, they physically move them right here to the plasma membrane of the neuron and release the acetylcholine into the synaptic cleft, which is right here. So right over here, we have a muscle cell, one muscle fiber. So I'm now drawing upside down. that's what we have happening so far. So at this point, the signal has been converted from from electrical, a change in charge, to chemical. Here's your signal right here. It's in the acetylcholine. So don't worry about all of this just yet. We can walk through the first little bit. So here are those myelinated sheaths that I talked about before. So all of those gates opening, the sodium and potassium, the individual sodium gates, the individual potassium channels, and the sodium potassium pumps. They're all in these little areas in between the myelinated sheaths, called the nodes of grandia. They are not underneath them. Well, they were a different function because nothing can get through there. So here's the end of the axon. So this is the end of the nerve cell. And pretty much what I've drawn here, hopefully the same thing. We have vesicles that contain, they're using little red dots to represent acetylcholine. These are the calcium channels, voltage gates. They change shape by a change in charge. So that change in charge comes, so it was originally negative here, positive here. Now it becomes positive here, negative here. That change in charge opens up the gates by changing the shape. That's all it does. It just changes the shape of the protein, which is enough to open it up, in particular to just this one molecule. Calcium comes in, and then it binds to the vesicles, brings them to the synaptic cleft. The vesicle membrane, which is made of the same stuff as the plasma membrane, they just fuse, and they become one, and that releases acetylcholine into the synaptic cleft. So this is the synaptic cleft right here, this space. This whole thing is the neuromuscular junction. So this whole area where they meet, where the nerve meets the muscle, that's the neuromuscular junction. This is the synaptic plate. And this bit right here, that's the motor plate on the muscle cell. We'll go through what happens in the rest of it when we get there. I'm not quite there yet. So, the acetylcholine crosses the synaptic cleft. I'm going to need more colors. I'm going to need another chalkboard, isn't it? So we're yellow, are we, for acetylcholine? Sure, why not? Let's use this chalkboard. Can you guys see this? Is that anywhere? So here's the axon terminus that I just drew over there. You got your acetylcholine. Side of it. Some of it's being released by the calcium. So the calcium ions come in. So they're fusing. And release. Underneath, we have sarcolemma of a muscle cell. Here's our muscle cell. One muscle fiber. So this space here is the synaptic cleft. This whole thing is the neuromuscular junction. Like everything I'm drawing here is part of the neuromuscular junction. I don't know what the good chalk is. Directly underneath the axon is the motor end plate. So what I'm drawing here is folds in the sarcolemma. So this is the plasma membrane of the muscle cell. Okay. So we've got little bits of acetylcholine coming out, coming down. Okay, so can't read last time. Probably over there. There are sodium gates. Always, always sodium gates. And that's why there's folds here, so you can have all kinds of sodium gates. I'm not going to draw them all. Just know there's a black sodium gates. The difference here, and this is really important, difference here, these sodium gates are not voltage gates. They're chemical gates. They're neurotransmitter gates. They're operated by molecules showing up and fitting in the right spot. And the molecule that has to show up, of course, is acetylcholine. Acetylcholine attaches to them and they open up, which means sodium comes rushing in. Because once again, like every other cell in an animal, there's a net negative charge in the muscle. There's less sodium in the muscle than there is outside. If there's too much sodium in any cell is going to be toxic. It also has sodium, potassium pumps, which are doing exactly the same thing here as they're doing in the nerve cell. Constantly removing sodium and bringing potassium in at a rate of 3 to 2, creating that net negative charge. So sodium comes rushing in here, which causes an action potential to happen exactly as it did in our neuron. So we have a change from change from negative on the inside, and positive on the outside, to positive on the inside and negative on the outside. And that happens all the way along in the same stepwise fashion as it did in the nerve cell. So we have our action potential going both ways, actually. But what does that mean? So what? Go to the next point. It's almost over. We're almost to the point that we started with muscles. There we go. This causes an action potential. That action potential moves down the T tubes that I mentioned yesterday. It was yesterday, wasn't it? I think it was yesterday. I hope it was yesterday. Those little openings in the sarcolemma, T tubes, that have the reservoirs on either side with all that calcium just waiting to do something. What is the something the calcium is going to do? What does the calcium do in muscle contraction? What's the role of calcium in allowing muscles to contract? It moves the troponin and the tripomycin out of the way of the actin, off the actin, so the myosin can hit it. Well, it can't get out yet. It's stuck in these reservoirs. But in these reservoirs, of course, are voltage gates. Voltage gates for calcium. And as the action potential moves along the muscle, along the sarcolemma, it goes down the T tubes, changes the net charge here, affecting these protein gates and opening them up. They open up, calcium floods out and goes over to the myofibrils, moving the troponin, or allowing the troponin to move the tripomycin off the actin filaments, allowing the myosin head to hit the actin filaments, and going through the whole prosperity that we discussed yesterday. So, continuing on, acetylcholine comes out into the synaptic cleft. So what I just tried to draw, this is much better. You have voltage-controlled sodium gates. Sorry, they're not voltage controlled, I lied to you, they're neurotransmitter gates. The acetylcholine attaches, there's a receptor site on the gates. Acetylcholine attaches, by attaching to the gate, it changes the shape, it opens up, sodium rushes in, causing the action potential. So we move along. Move along the surface of the muscle to the T tube, where it comes down the T tube and opens a voltage gate, which allows calcium out. Once the calcium gets out, the muscle contraction can start. So we've gone from transmitting a signal into a neuron, creating an action potential, converting it to chemical, creating it back to electrical, to starting the muscle contraction process. As soon as those calcium ions get out, it all happens. Because they're going to automatically remove the troponin and tripomycin. So along the T tube, the action potential activates the calcium voltage gates, releasing the calcium over the bowel, excuse me, myofibrils. This starts the cross-bridge process leading to the contraction. Meanwhile, something else has to happen now, because the gates that let the sodium into the muscle cell aren't electrical, so the passing of the electrical current isn't going to revert back to a negative charge because of the potassium pump, because it wasn't electrical to start with, it's chemical. So all these gates are now open. They've been opened by acetylcholine, right? Now they have to close again. Well, it just so happens the cell is full of acetylcholinesterase, which is an anti-acetylcholine, if you will. It goes up to the acetylcholine that's attached to those now open sodium channels and degrades it. And so without the acetylcholine in the receptor site, the channels close. And that's what stops the sodium from coming in, and that stops the action potential from continuing. And the sodium pump, the sodium-potassium pump, resets that negative charge. So within the muscle, along the surface, underneath, along the cytoplasm, the sarcolemma, the plasma membrane of the muscle, you have this acetylcholinesterase, which degrades it. And those component parts go back into the nerve cell to make more acetylcholine to be packaged up into vesicles and await the next action potential where the calcium is going to come in, bind with the vesicle, take them to the end of the axon, release them into the standard pattern, so on and so forth. So when the sodium is blocked from entering the cell, combined with the gates, the pumps I just mentioned, the cell is repolarized. It goes back to a net negative charge. So acetylcholinesterase degrades acetylcholine, shuts the gate, the potassium pumps are bringing in potassium, taking over the sodium. We only have one thing to deal with now. Do you have any idea what that might be? It hasn't been resolved. We have a contracting muscle that has been polarized. That's why it's contracting, or depolarized. It's been repolarized. But it's contracting because of all of this calcium, right? So the calcium is still out. It hasn't been put back into the reservoir yet. Without which your muscle is never going to uncontract. Or decontract, is that a word? Not contract. Fortunately, those reservoirs also have a calcium pump, which is driven by ATP, so it can move across, or it can move against the gradient. So it's, as soon as the calcium gets released, there's another one-way pump that starts pumping it back in. So as soon as the action potential stops, the voltage calcium gate shuts. It doesn't let calcium out, but the one-way pump that's bringing it in keeps going and starts to pump it out of the myofibril back into the reservoir next to the T2. And when there's no more calcium over the actin, the tripomycin goes back into place, and the myosin can't hit it, and the muscle relaxes. So there's no more cross bridges anyways, no more power strokes. These things don't work anymore, sorry to say. But that's the whole process. That makes sense. We've gone from firing a nerve, how a nerve works, to how that nerve makes the muscle work. And that's an organ functioning at the organ level. So what are the emergent properties here? We would give feedback loops. Hormones can turn on and off those functions. We have signaling, that ability to have physical motion. This is now a product of the organ level organization. You don't get the motion of a muscle by the muscle alone. The muscle can't operate on its own. It needs the nerve to trigger the action potential that allows it to happen. So that's why the combination of tissues is important. So that movement of muscle, or the contracted muscle, only happens at the organ level. You also have at this level creating and depolarization and repolarization, allowing a signal to be transported across the entire body. It's instead of just locally within cells. Wow. There must be a button there. Don't you know that? Oops. And of course, feedback loops here, I mean, not just blood pressure, which I mentioned before, as a feedback loop of the organ level. Feedback loops here are also the cell, the nerve cell, the potassium working in conjunction with the sodium, but the potassium wouldn't open if the sodium didn't open. So this is a feedback mechanism, a balancing mechanism. So that's all I want to cover today. Probably not. And for tomorrow's exam, I will show you, I think there's another. No, unfortunately. I will post the one I just showed you. Unfortunately, it doesn't really show you what's happening at the neuromuscular junction. I'll see if I can find another one that does. I'll post that too. Okay, so let's take a break, and then after the break we can have questions about the exam, and then that's it. No questions, we'll stop, and you guys go home and get it. I will tell you a little bit about the structure of the exam first, but first break.
Okay, so after class, because I didn't attend the first meeting, and also this means that we will have the notes, right? I mean, this week's lecture notes you all have it, right? Yeah. I mean, after class, can I take a picture of it? Yeah. Okay. So.
Verify. I look at it. That's gonna make us think I'm gonna go back again. I was just, I got up, and I went back to the bed, and I just sat down in my bed. I was like, amazing. So literally making my life on your sleep. Whatever you're doing, you're just sitting there. And I woke up this morning at like 6 and I thought it was raining for some reason, so I didn't. ちょっと休憩してからしよう。super eager to go. Yeah, I did have her Snapchatting so I like I was like, and then I woke up and I wasn't so then I came back and I was like sleeping. I was like after something I felt bad. I think I spelled super wrong because I was half asleep. I was like, what? I think I said no. So I spelled it like N-A-D-A. So wait. Swallow. I wasn't sure what happened. Did she send you that thing? Like, she sent you sunscreen. Oh no, I didn't wait for any of this. I was exhausted. I'm tired. I went out and watched an office. Watching the phone. The middle of the bed. I get to just hear your phone. And I thought you guys I know. I wanted to do it like this. I think it was really good. Well, that made more sense than what I wanted to do. Yeah. We usually do that. That's what I do every weekend. Travel.
Okay. I just posted a couple more videos, one of the neurons and neuromuscular junction function. Right to the far end of the myofibrils. So check those out at your leisure, and it will certainly help with studying for the exam. So speaking of the exam, it's tomorrow. The format I normally would do pretty much the same thing. It's a multiple choice, maybe even some multiple choice, like some of the questions I've shown you where there's more than one right answer. I usually do at least one kind of essay. I might even give you a choice depending on what I'm doing. And word match, but I guess I want to get a sense from you guys if you want to do the word match. Because what I found, contrary to most of the classes where generally people do well, last time some people did really well, but some people did very poorly. It was very biomotor. Either you did great on it or you did very poorly on it. Whereas normally there's a bit more of a distribution in between. So I want to get a sense from you if you want a word match. If not, I can do something else. I could add more multiple choice or more short answer or what have you. So who would like to see the word match kept in? Your hand up. That's 2, 3. Who would like to see the word match removed from the exam and replace it with multiple choice or short answer? 1, 2, 3, 4, 5. Okay, I think that wins. Democracy is a wonderful thing. Okay, so there won't be a word match. I'll have to come up with a bunch more multiple choice questions. I hate having to come up with multiple choice. You have no idea how hard it is to come up with 4 or 5 incorrect answers that sound plausible. Like, it's easy to put the right answer in. I'm gonna have something else that sounds kind of like it could be right. So it's really a lot of work. So are there any questions? And once again, I don't have any formal prepared because what we just did it. So, but I will certainly entertain any specific questions at all. I can go through something again if you wish. Otherwise, go home and study. Enjoy your day. Yeah.
Are we gonna have to like draw anything, like any of these, like?
I don't know yet. Instead of an essay question, I might have a diagram. I like doing the diagrams. If you can draw something out, it tells me that you really understand it. And that's what I'm looking for. I'm trying to think what I would do if I was to do a diagram. That's much more complicated than I would expect you to be able to draw. And it's pretty messy. If I saw that, what the hell is that? I may have you label a diagram. Yes, that is most definitely something I will do. What that diagram is off the top of my head, I don't know. But I could show you something like that and say, okay, like what's this gate? Where's the axon? Basically, I'd have arrows pointing to certain features, and you Or I could just show a general cell do the same thing. Show a general cell do the same thing. Anything else? Are you just dying to get out and enjoy that beautiful day out there? That,
So I was a bit confused when something said like extracellular and non-cellular matrix. Is that like there's no cells in it at all?
no, it just means the matrix itself isn't made of cells. So there's cells in it, but there's a bunch of other stuff that isn't cells. Like all of those fibers I mentioned, the collagen, the elastin, those fibers themselves aren't cells. They're made by cells, not in the matrix, but they themselves are not cells. Just like bone. But most of what you see in bone is not cellular. It's just calcium. A bunch of other stuff. Calcium bicarbonate. Anything else?
Is there some difference between pluripotent sites
And Schwann cells? Yes, but you don't have to know it. You can use those interchangeably. That's fine. Yeah. At this level, absolutely. There are, but don't worry about it. At this level, they're the same thing.
Would you be able to explain the endosymbiotic theory again?
Yeah. Basically, the endosymbiotic theory is the notion that you've got eukaryotic cells from a combining of some prokaryotic cells, either eubacteria and an archaeobacteria or two archaeobacteria. It doesn't really matter which. I don't expect you to know a lot about bacteria because that's not what we're studying. But do know that the first eukaryotic organisms were single celled, and they came from one bacteria engulfing another but not consuming it. And over generational time, billions of years, that relationship became symbiotic. So an example would be if you had a bacteria that was like mitochondria. So it's just this little powerhouse. It's tiny. It's I'm trying to remember which was bigger, eubacteria or archaeo. I want to say eubacteria, I'm not sure of that. Archaeobacteria, I think, are smaller, so it gets consumed by eubacteria, but it doesn't get eaten. But it's still generating power. And so it's producing ATP that now the larger cell is using. And it gets used to that. It's like, hey, this is a great idea. The smaller cell gets protection. It gets everything it needs, shipped to it. All the food and stuff is all taken care of by the larger cell, and it provides for that larger cell power. And say over time, it stops being two different kinds of cells, because that would have originated one cell living inside another cell. They would have gone through synthesis independently. They could still be living together, like lichens do. They would go through synthesis and cell division separately, but over billions of years and generations, that smaller cell becomes incorporated as an organelle in the larger cell. That's how you get your first eukaryotic cell, because that smaller cell now has its own membrane. And that's where you start all of these other little organelles that have the little membranes. That's where it starts. So we can move from just prokaryotic cells to eukaryotic cells. But yeah, that was billions of years in the making. Is that helpful? Any other questions? Going once. Going twice. God. Then go enjoy the rest of this beautiful day. Do take some time to study. They say I just posted 2 more videos. So we'll help elucidate everything I talked about today. Okay. If the links don't work, email me. And if you have any questions while you're studying today, email me. That's absolutely fine. I do believe I posted my email on the website. Yes, I did. So, if you have any questions while you're getting ready for the exam tomorrow, just shoot me an email. I'll answer them throughout the day.
I'm usually this Monday. Yeah. I wonder if you the two paragraphs on this, right? Yeah, from here. Okay. Yeah, so it's from this page, right? From this page. From here. Yeah. You wanna to here, right? Actually, yeah. Anything else? From here to here, right? Yeah. Okay. I realized that in some classes the instructor does also some exercise questions, right? Did you drop the no problem? No. No? Okay.